Hello everybody, I am Cristiane Taniguti and I'm here to introduce to you the work comparing genotype calling software performance in genotype-by-sequence data of outcrossing population based on genetic maps quality. The high-throughput sequence methods produce millions of sequence reads that needs to be processed with bioinformatic tools before being applied in genetic research. For each step of the process include the alignment of reads, SNP identification, and genotype calling, several tools are available, all with different methods and parameters to be selected by the user. Changes in a single parameter in the pipeline can cause downstream consequences in the quality of the analysis. Because of the genetic properties of the meiotic events are well known, linkage analysis can be used to identify low-quality markers, as shown in the figure. The left side shows a linkage map built without genotyping errors, and the right side with genotyping errors. We can see that in the presence of these errors, there is a wrong number of recombination event, inflated linkage map size, and distortion in the expected color pattern in the recombination fraction heat map. Therefore, good quality genetic maps validate all upstream procedures and can help to identify the best combination of software and parameters. Here, we present the Reads2Map workflows to build linkage map from sequence data of experimental F1 outcrossing population, testing combination of upstream tools. The workflows are written with workflow description language, also called as WIDO, which offers a comprehensive structure and metadata for each step, making it easier for users to adapt specific parameters. There are two main workflows available, one for empirical and another for simulated data. Both use BWA to align reads, GATK and Freebase to perform the SNP calling, and UpDog, PolyRed, and SuperMassa for genotype calling, and OneMap for linkage map building. WIDO also allows an interface with Docker and Singularity containers to increase reproducibility, facilitate access to diverse software, and use in high-performance computing or cloud service environment. The crawl-run software can be used to run the workflows with a single command. The final workflow output is the input for Reads2Map app, a Shine app which allows interactive visualization and exploration of the genetic maps produced and selection of the best pipeline. Here you can see an example result from the simulated Reads2Map workflow. We run the workflow using mean read depth of 20x with 5 repetitions, with and without segregation distortion, and with counts from the BANA alignment files and VCF files. The genotype probabilities from each software and also a global error rate of 5% were used in the OneMap HMM chain to estimate the genetic distance. The graphic shows the final map's number of markers and total size for each combination of SNP and genotype call software. 2 extra filters were tested to reduce the number of outlier markers, a minimum genotype probability of 0.8 and the removal of the non-informative markers. The horizontal red line shows the real map size of 38 centimorgans. We can see that after filters, most of genotype calling methods can build maps close to the expected size with GATK markers. A similar graphic is presented with results from empirical Reads2Map workflow. We run the workflow using GPS data of a rows deployed outcrossing population sequence with mean depth of 94x. Here we can see that the filters could remove some of the outliers. However, compared to the simulated data, fewer methods could reach the expected map size of 38 centimorgans. Evaluating the show graphics and others available in the Reads2Map app, we could select 2 pipelines that better reproduce the expected number of recombination breakpoints according to meiosis properties. The figures show number of recombination breakpoints by individual, the linkage map draw, and the recombination fraction matrix heat map generated by the selected pipelines. The main conclusions and perspectives of this work is that the combination of WIDO, Docker, cron and Shine structure makes the Reads2Map multi-platform practical, user-friendly, and reproducible. Filtering markers by the genotype probabilities and informativeness help to remove outlier markers that distort the genetic distance. The usage of global error rate of 5% and the 1-map 3.0 HMM chain midpoint approach result in genetic maps with a size closer to expected. However, in some cases, it can underestimate the number of breakpoints. Next, Reads2Map updates will be focused on adding map-only for downstream linkage map construction of autopolyploid species. By now, that's it. I would like to thank you all project members and the funding agencies.